Hi everyone, I'm Zixuan Lan, a first-year PhD student from the Philip Avada Lab at University of Toronto. It's a great pleasure here to present my project to all of you. Thanks GLBIO for this opportunity. Today I'm going to present my PhD work so far. Tracking the elusive paths of cancer stem cells, a study on their trajectory and implications in cancer evolution. Stemness refers to the ability of a cell to undergo unlimited self-renewal, to differentiate into mature cells, and to generate heterogeneous populations. Some cancer cells display stemness and are considered as the main drivers for tumorigenesis and metastasis. They are the so-called cancer stem cells. They also upregulate many of the stem cell signaling pathways, including Wnt and Notch. Cancer stem cells might originate from stem cells that gain oncogenic mutations or cancer cells becoming stem-like. In liver tissues, there are bipotential ovar cells, which can differentiate into hepatocytes or bile cells. Because of this property, people reason that this might be the cancer stem cells in hepatocellular carcinoma. Therefore, we are interested in when and how cancer stem cells involve in the liver tumor population by building developmental references of liver cells with single-cell transcriptomics. We start with collecting single-cell RNA sequencing data from different sources, including fetal cells, stem cells, cancer cells, and normal cells. In particular, our liver cancer dataset is composed of tumors as well as adjacent normal cells. This makes correction of batch effects easier, as we no longer need to deal with another data source, so we are minimizing our variation. We are also doing one patient at a time to reduce the variance among patients. We deal with batch effects using FastMNN, which is a faster version of the mutual nearest neighbor approach. Then we project our integrated data onto FATE. FATE stands for Potential of Heat Diffusion for Affinity-Based Trajectory Embedding. It is a nonlinear dimension reduction method, and it offers visualization in lower dimension. Here is the result of FATE applied to our data. We have a combination of fetal cells, tumor cells, stem cells, and normal cells adjacent to the tumors. Actually, what you can see from this figure here is a trajectory that starts from stem cells and proceeding to the fetal cells. What is interesting is that there's a branching structure here at the end of the fetal cells, and leads to two different paths, the tumors and the normal cells. We also see some cancer cells overlapping with the stem cells, and that's when we start to hypothesize that these cells might be the cancer stem cells we are interested in. To look at these cells more closely, we separated the cancer cells into 4 groups based on their locations onto the FATE map. The cells closer to the stem cells are the stem-like cells, the cells at the branching point are the we call middle ones, and the cells at the very end of the path are the so we call upper ones. I circle the green dots here as they are too few and too small to see, and they are the stem-like tumors we are mostly interested in. Next, we compare our annotations with the labels assigned by UMAP, which is another dimension method. And what we can see here is the stem cells and stem-like tumor from the same cluster in the UMAP. And that's the cluster 2 in figure 3. This sort of serves as another layer of validation that these two groups have similar expression profiles, and that the cancer cells might be stem-like. Then we look at the significantly differentially expressed genes in the two groups, and that's what we are trying to show in figure 4. If we are only looking at the x-axis, it shows the fold changes of the markers in the stem-like tumors. Similarly, the y-axis shows the fold changes of the markers in the stem cells. And if we look at the fourth quadrant together of the two axes, we can see that there are many genes that are highly expressed not only in the stem cells but also in the tumors, and that's how we obtain our list of highly co-expressed genes. Then we perform gene set enrichment analysis on our list of genes, and we find out that they are associated with ribosomal function and respiration. This tells us that the cells experience high activity of protein synthesis and ATP production, which is an indicator of high metabolism in the cells. We ensure that these high activities are not because of the cells are in S phase, because of their cell cycles, as they do not highly express any of those genes. And to summarize, we aim to characterize stemness in the tumor population by creating a model that imitates the progression of cancer cells between differentiation stages. From a single liver cancer patient, we identify some cancer cells clustering with the stem cells. The stem-like tumors share a variety of upregulated genes with the stem cells, and these cells experience high cellular growth and may display some extent of stemness. Next, we will try to further explore stemness in our stem-like tumors. We will also start to look at other liver cancer patients to identify more tumor-like stem-like tumor cells and to obtain a more comprehensive gene signature for cancer stem cells. Then that's the end of my presentation. Thanks everyone for listening.